Oh, my bad. I forgot you guys didn't understand parcel tongue. Anyway, hello everyone. We are Group 4 and we will be talking about how proteomics has helped us understand the venom composition of snakes, especially the venom of the spectacled cobra.
From the subcontinental Indian countries, the Naja Naja is responsible for over 6,000 deaths a year. Yes, this highly venomous snake has a distinct wide black band below the neck and half rings on the cobra's hood. Its smooth scale body is covered with a yellow-white spectacle pattern, giving it the name the spectacled cobra. And it can grow up to 2.2 meters. With a lifespan of 23.9 years, the Naja Naja cobra is mostly found in wild forest and cultivated areas where it feeds on lizards, frogs, and diseased rodents. Naya Naya species has both a negative and a positive impact on human health. Its venom is both paralyzing and lethal. However, the cobra's venom has medicinal properties and is considered as a potential source of anti-cancer drugs and painkillers. From the genus Naja, the Naja Naja venom is medically important in South Asia. This led to the study of the venom proteome to identify the properties of the venom with anti-cancer properties using methods such as mass spectrophotometry, SDS page, data mining, and high-performance liquid chromatography. Merriam-Webster defines proteomics as application of molecular biology, biochemical, and genetic techniques for the analysis of protein function, interaction, and structure organized in databases for data application. Simply put, proteomics details the venom composition both quantitatively and qualitatively, which provides a better understanding of the constituents to facilitate both drug manufacturing and development.
In most of the reports studying the venom composition of the Naja species, we see a pattern of methods used to analyze the composition. We start with the fractionation of peptides present in the venom using chromatography and electrophoresis. A wide use of reverse phase high performance liquid chromatography or RP-HPLC for fractionation was seen in the studies, while some other methods such as ion exchange chromatography, size exclusion chromatography, and affinity chromatography were also used. RP-HPLC fractions are subjected to SDS page or two-dimensional gel electrophoresis or two-dimensional differential gel electrophoresis for better separation of low molecular weight proteins, and the protein bands are stained and scanned to acquire an electrophoretogram, which can be used to find out the approximate molecular masses of venom proteins and the relative abundance of each band. The protein gel band of interest can then be separated out by different chromatographic techniques based upon the physiochemical nature of polypeptides and digested using MS-grade trypsin solution, followed by analysis of the purified peptides using tandem mass spectrometry. Various kinds of tandem mass spectrometers were used in the papers we analyzed, such as tandem time of flight and liquid chromatography tandem mass spectrometry. The data from the mass spectrometer is then extracted and processed by various softwares and provide us with the sequences of proteins in the venom via de novo sequencing. The protein sequences are homology matched against databases containing known peptides such as the UniProt SwissProt protein database, non-redundant NCBI database, or in-house databases. Sequence databases are the core of bioinformatics as they contain gene, mRNA, and protein sequences documented to date and allow us to run our results through multiple sequence alignment and find the identity of the unknown sequences. Certain steps would of course vary from experiment to experiment, but these are pretty much the basics of how we use several proteomics techniques to acquire the protein identities.
Proteomic analysis of Naja Naja venom in several studies have allowed us to identify that there are two proteins most abundantly present in the Naja Naja venom, the non-enzymatic three-finger toxins and enzymatic phospholipases A2. These show specific binding to postsynaptic nicotinic acetylcholine receptors at the neuromuscular junction in skeletal muscles and cause the blockage of neuromuscular transmission. While the other toxic proteins of cobra venom, such as snake venom metalloproteases and cobra venom factors, provoke local tissue damage and necrosis. A revealing 2018 study compared the venom of Pakistani Naja Naja with previously reported Naja Naja venom compositions and found out that it contains a much higher abundance of alpha neurotoxins than Naja Naja venoms of India and Sri Lanka. This made the Pakistani Naja Naja venom more lethal than the others, as alpha neurotoxins are highly potent postsynaptic neuromuscular blockers that cause paralysis. Most of the antivenom available in Pakistan is imported from India, which is targeted towards the Indian Naja Naja, hence it is not as effective as it must be, and this is highlighted in the study. This was a particularly daunting revelation as it suggests that the victims of Naja Naja envenomation, especially in the rural areas of Balochistan, Punjab, and Sindh, pay tens of thousands of rupees for a partially effective antivenom. This finding can help us synthesize antivenoms with antibodies that neutralize these alpha neurotoxins more effectively and which can be easily accessible and affordable to the poor population of Pakistan.
In another study, the blister fluid was collected from 6 patients in India bitten by Naja Naja and 2 other species of snakes. After a proteomics-based analysis of the exudates, a total of 805 different proteins were identified using a multiple database search algorithm known as sequenced proteome discovery. Informatics analysis of the proteomes identified the top biological response categories of these proteins. The study supported the concept that aseptic removal of blister fluid may reduce the extent and time dependence of venom-induced tissue damage, as the venom present in the blister fluid could further diffuse into the tissues. Lastly, a study compared the venom proteomes of cobra venom from Southern India, Eastern India, and Western India and showed that 12 proteins are common while 21 proteins were exclusively present in Southern India Naja Naja venom. Therefore, qualitative and quantitative differences in the venom composition among these same species of cobra venoms from 3 different locales of India suggested variations in clinical symptoms and severity of pathogenesis following cobra bite in these regions. This study, along with many other proteomic-based studies, support the same hypothesis that despite the cobra belonging to the same Naja species, the venom composition does differ based on where it is geographically located.
Antivenom is the only treatment for snake envenoming, and now we can make specific antivenom for specific venoms based on composition and save hundreds of lives with a single injection. Antivenom consists of polyclonal antibodies that can neutralize multiple toxins in snake venom, and they are mostly produced in experimental animals that are injected with repeated small doses of snake venom. The antivenoms can be categorized as monovalent, against one species of snake venom, and polyvalent, against various species of snake venom. Mostly, antibodies in antivenom prevent the interactions between toxin and target tissue by binding to the toxin and lead to confirmatory change of pharmacological site, therefore neutralizing the toxin. Surprisingly, the horrific snake venoms also have valuable natural pharmacological action that contribute to medical research purpose and drug discovery. Snake venom isolated from Naja Naja together with other snake species possess anti-cancer properties that are toxic to the cancerous cells and therefore destroy them. Also, studies suggested that Naja Naja snake venom demonstrated anti-HIV activity. However, both of these drug discovery related to Naja Naja snake venom are still in development. Several drugs developed from snake venom proteins have been approved by FDA. In 1975, captopril was the first successful and reputed example of a drug developed on the basis of snake venom component. It is used to treat hypertension and cardiovascular disease by preventing constriction of blood vessels, which raise the blood pressure. Integrelin inhibits the final step of blood clotting process, which treats patients with acute coronary syndrome to reduce the chances of heart attack or even death. If you want to be gorgeous, you probably need some venom on your face too. Alpha neurotoxins, named Walgreen, was used in the development of anti-wrinkle cosmetics.